Hello, everybody, and welcome to the 2025 California Invasive Species Action Week. Our speaker for today is Dr. Mark Hoddle. He is the head of the UC Riverside Applied Biological Control Research Lab. He's held that role since 1997. He's primarily involved in the study of pest problems, where biological control could be a successful approach. The location, release, and evaluation of natural enemy impacts on population growth features very strongly in his research. He looks at biocontrol agents, conducts regular studies in the field, and when necessary, aspects of both pest and natural enemy biology and behavior are studied in the laboratory as well. He's also the director of the Center for Invasive Species Research. So, Mark, go ahead and
Okay.
let's share your slides.
Super. Right. Thank you, Randall. Thank you, Julie. And good afternoon, everyone. Right. Let's see if I can do this without making any mistakes. Oops, share that. I've already made an error, maybe. There we go. How are we looking on your end, Randall?
It looks good right now, yes.
Excellent. Okay, well, thank you for the invitation today, and I'm happy to be talking to you about a new area of biological control we've been developing down here in my lab and with some colleagues overseas. And we're going to be talking about proactive biological control of invasive species, and the focus is going to be on mainly invasive insect problems. And in the opening slide here, you can see Canary Island date palms that have been devastated by the South American palm weevil in San Diego County, and this is one project we've been working on recently. So I'm going to set the stage. We're here for invasive species, and I think getting us all on the same page so we know what we're talking about is important. And I've taken this page out of the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services, which was published in 2023. It's available as a PDF. It's a beautiful document, and it's worth reading through. So native species that we are all familiar with, that they're found within the native range of that organism. The next organism that they list here are alien species. And an alien species has a presence in an area outside of its native range, and this is often attributable to human activity. It hasn't spread into this new area on its own. So an established alien species is defined as a subset of those alien species that are self-introduced, but they are now self-sustaining and they're spreading throughout their new range. And this is where we are most interested in management, are invasive alien species. These are defined as a group of non-native organisms that have established, they're spreading, they're reproducing, but they're causing some sort of ecological, economic, social health problem. And this is often referred to as this phase is often referred to as the biological invasion. And it's going to be a multi-step human influence process that has moved these non-native species into new areas. They have now established, spread, and they're causing problems that require management. And it is this group of non-native organisms that we're going to be focusing on in this presentation today. Okay, so you're probably familiar with this term invasive species and that they cause some types of problems. But, what exactly are the issues here? Well, a lot of this has been summarized in this document, and it was compiled with the assistance of about 86 global experts who have experience with managing invasive species, and also had additional input from an additional 200 colleagues. And it is an in-depth document. They cite about 13,000 scientific publications. And some of the interesting things that I just picked from this was that their conclusions suggest that about 60% or so of species extinction have been driven in part by invasive species, and they cost around $423 billion a year to manage, and about 92% of that, or $390 billion, is actually managing the damage that is caused, and about 8% of it goes to keeping them out of the areas in which they are not wanted. So there is a disproportionate amount of money spent on managing these problems as opposed to mitigating the factors that lead up to the invasion process itself. Okay, so, well, what are some of these problems? Well, more than 37,000 species are now considered to have established outside of their native ranges, but not all of them cause problems. A handful of them cause problems, and those are the ones that we are obligated to manage in some way because they're causing some sort of economic, environmental, or health problem. So about 3,500, approximately 10% of those 37,000 species cause some type of negative impact. 16% of known extinctions are attributable solely to those invasive species. And interestingly, the rate of spread is continuing. This is something that is not slowing down. It hasn't plateaued. And about 200 non-native species each year are being recorded in new areas where they previously did not exist. So these non-native organisms that are causing problems impact our economies, affect our food and water security, human, plant, and animal health. And the global cost in 2019 was estimated to be about $423 billion. So we're talking about significant sums of money here. Interestingly, these invasive species can also contribute to marginalization and inequity. So for example, an invasive weed or a fish, an insect may establish somewhere. It may reduce agricultural productivity, for example. And in the photograph that you can see up the top here of this lake that's being choked with this floating aquatic waterweed, transportation across water surfaces is now being impeded. And the impacts often fall disproportionately on young kids and females who end up having to carry the brunt of the load when it comes to harvesting the food, planting the crops, or dealing with the management of these invasive species. But what I'm going to be focusing on now is the last part of this title, the invasive alien species and their control. That's what we're going to be focusing on for the remainder of this presentation now that we've set the stage on what we're actually dealing with. Okay, so what control options do we have for managing these invasive species? Prevention, preparedness, and control are all management options that are available for us to use. Prevention often occurs pre-border or at the border, or maybe even at the post-border level. And these are the basic biosecurity practices that are enacted to either stop something coming into the country before it leaves the country of origin. So for example, these may be inspections of fruit that have been harvested, they've been packaged, and now they're going to be sent to California. Inspections may happen at the border at the time those goods arrive, or possibly immediately post-border once those products have landed, they've been moved, and then they may be subjected to another set of observations. Preparedness, this is something that interests me a lot. We may conduct what we refer to as horizon scans, where we are looking out into the broad global community here to identify known threats, maybe new pest problems that are beginning to emerge that could possibly come to California. So we're getting prepared for something ahead of time. And a good example of this is spotted lanternfly. This is an agricultural pest that is native to China. It is established widely on the east coast of the United States. It has now established populations in the Midwest, and it is highly likely that at some stage this grape and nut-feeding pest will arrive in California. It is likely to establish, spread, and cause some type of economic problem for us. Something that the California Department of Food and Agriculture does a lot of is early detection of incipient populations. If you can find a new pest population early enough in the invasion process when it is still small and relatively localized, you may be able to contain it and eradicate it. And finally, the last step is once prevention, early detection, and eradication have obviously failed, these pests are now established and spreading widely, causing some sort of problem that we need to manage. We now need to think about our control options. These could be physical, putting up sticky traps, for example, to catch pests. A lot of you are familiar with chemical control practices. We use insecticides, herbicides, and fungicides to control problematic organisms. But what I would like to focus on now are the biological options that we have available to us, our natural enemies. And we're going to be focusing on biological control for the remainder of this presentation. So there are several different types of biological control. You may have heard of classical introduction, importation, or inoculation biological control. This is something my lab focuses on. So an invasive organism that comes to California and establishes here, it's not native to this part of the world. It has originated from some other part of the world. And part of my job as a biological control practitioner is we practice something that we refer to as foreign exploration, where we go back to the home country or the home range of the invasive pest that we're trying to control, and we look for the natural enemies, the parasitoids, the predators, or maybe insect pathogens that may exert some level of control over that target pest. Those natural enemies may have potential for importation and use in California for suppressing pest populations to levels that are less damaging. Augmentative biological control is where you may mass rear or purchase mass reared natural enemies and you release them into an agro-ecosystem, for example. So one example of this are the augmentative releases of a scale parasitoid called a Phytus malinus, which is used in citrus for the biological control of California red scale. Augmentative biological control releases augment or boost existing populations of natural enemies that are already present, but their numbers may be a little too low to afford immediate or highly impactful control of the target pest. Inundative biological control is sometimes used in greenhouses, for example. You may have a valuable vegetable crop or a horticultural crop of important, high economic importance. So you may purchase a lot of mass-reared natural enemies and you flood the greenhouse, for example, with those natural enemies, say whitefly parasitoids, for example. And essentially you're trying to overwhelm the system and the natural enemies are being used, I guess, as a type of biological insecticide to try and drive those pest numbers down to much, much lower levels than what they're currently being observed at. And finally, we have conservation biological control. This is a practice where you may have an orchard, for example. There are resident natural enemies there, but you want to improve the living conditions for those natural enemies. You want to conserve those populations and maybe boost them by providing resources that may be missing in that orchard. So, for example, planting flowering cover crops is a good example of conservation biological control. Those flowers provide pollen and nectar to natural enemies and parasitoids, for example. Females may use the nectar for energy to fly around looking for pests to parasitize, and if they feed on the pollen, that may provide the protein that they need to mature more eggs. We're going to add to this standing, well-accepted list of biological control practices a new option for us now. And I'm referring to this as proactive biological control. And this is going to be the discussion for the remainder of this talk. And we're going to investigate this idea that if we use horizon scans to identify invasive species that are floating out there that are encroaching upon California, could we initiate a biological control program in advance of the anticipated invasion of that insect pest into California? Could we screen natural enemies ahead of time and get approval for their release before that pest shows up here in California? Often, our biological control programs are what I refer to as reactive. The pest invades, it spreads, it causes us problems, and then we think about developing a biological control program for that pest. And during that time frame, years may have passed, the damage is accruing, the biological control program is initiated, and then it takes years more of time to find those natural enemies, screen them for safety in a quarantine facility, get permission to release them, and then finally establish them in the field. Can we cut out a lot of that time and initiate a biological control program much earlier than we traditionally do? And that's what we're going to be discussing for the remainder of this presentation. Biological control, in my opinion, is a planned invasion with a natural enemy to control an unplanned invasion, which is the incursion of the pest species that we don't want here, but we now need to manage. So biological control is the use of host-specific classical biological control or an introduction biological control, where we bring the natural enemies in from overseas to target the pest of concern here in California. This is what I'm discussing. We're not going to be going over augmentative, inundative, or conservation biological control, we're going to be focusing on classical or introduction biological control. It's referred to as classical biological control because this was the first practice, the classic approach to managing these invasive pests was to bring in their host-specific natural enemies from the country of origin where the pest originated from. Okay, so we use host-specific natural enemies to reduce high-density pest populations to less damaging levels. And our natural enemies are typically predators, as you can see here, the Vidalia beetle sitting on top of a cottony cushion scale. Parasitoids in the middle, that's Tamarixia radiata that we imported from Pakistan for the, biological control of Asian citrus psyllid. Or sometimes we may use pathogens like fungi or viruses to control some of these invasive insect pests. So classical biological control is most often targeted invasive insect pests, weeds, and to a lesser extent, vertebrates. A good example you may be familiar with has been the biological control of rabbits in Australia, for example, with the myxoma virus and the rabbit calesi virus. Okay, so the process for a classical biological control program involves the following steps. Foreign exploration in the native range of the pest to look for natural enemies that may provide some level of population regulation in the native range. We bring those insects back to California with USDA APHIS permits, and we take them to the quarantine facility, which is a secure facility that we have here on campus. And then we do a couple of things. We look at what we call host range. That is, how many non-target species can the natural enemy successfully attack and reproduce on? Then the second phase of that evaluation is looking at host specificity. And this refers to, well, if the natural enemy has a choice of several different species that it can use for food, what does it prefer to attack? And ideally, what we want to see in our safety testing in the quarantine facility is a natural enemy that has a very narrow host range. It only attacks a limited number of species, and it is very host specific. That is, when given a choice, it really prefers to attack the target pest. Those characteristics help us in a couple of ways. It helps ensure that the natural enemy is likely to be safe when we release it into the environment. That is, it's going to pose negligible risk to non-target species. And because it's focused on the target pest, it's going to put a lot of effort into finding that preferred prey or host species. And if it's linked tightly to the pest in that way, ideally it should provide us high levels of control because it's either going to find something to feed and reproduce on, or it's going to starve and die. Those are very strong selection pressures that will be acting on a successful natural enemy species. So we finish up the safety testing, we prepare a report, it goes off to the USDA APHIS for review. The North American Plant Protection Organization also has a panel of experts that will review these data. They'll say, yes, the data look good, or maybe those experiments could do with a bit of improvement, or maybe you should consider testing these additional species. And then pending on those decisions, you may or may not be issued permits that allow you to move the species of your natural enemy out of quarantine into the environment for release. If you get the permission to release, you begin the mass rearing, you release the parasitoids, then you evaluate the rates of establishment, spread, and impacts. Well, California's done a lot of this. You may be familiar with this insect. This is the glassy winged sharpshooter. We have successfully controlled it with this parasitoid, Cosmocomoidea, formerly known as Gonatosaurus ashmedi, a parasitoid native to the southeastern United States, where glassy winged sharpshooter is native to, and it parasitizes the glassy winged sharpshooter eggs. And you can see the exit holes here where the parasitoid has emerged. Other successful examples globally include cassava mealybug, Phenacoccus manihoti, which has been controlled here with the cassava mealybug parasitoid. The pest and the natural enemy are native to South America. The pest itself is invasive in Africa and Southeast Asia, where cassava is a major carbohydrate crop. Now, this project I love. Cottony cushion scale was successfully controlled here in California with the Vidalia ladybug. You can see the little red ladybug down there in the bottom right-hand corner. The pest is native to Australia. It invaded California in the 1880s. It caused an immense amount of damage to California citrus. Arthur Cobelli, with assistance from agricultural entomologists here in California, imported the Vidalia beetle from Australia, and it provided very rapid control of this agricultural pest. Vidalia cottony cushion scale is also a major pest of conservation importance in the Galapagos Islands, and we were part of a team that evaluated the safety of the Vidalia beetle, which had been used to control an agricultural pest, for control of cottony cushion scale in the Galapagos Islands, which was killing off a lot of native plants that endemic insects and birds used for food. I love this project because not only were we working in the Galapagos on this, but it was a beautiful bookended project. We took an agricultural pest solution and used it to solve a problem of conservation importance. And the host specificity of the Vidalia beetle is extremely tight. It's highly focused in on the cottony cushion scale. And as a result, we got permission to release the beetle, the first biological control program ever run in the Galapagos Islands. And we got outstanding levels of control, similar to what we saw here in California. The plants have recovered, the native birds and the native insects that were relying on those native plants like Darwinia thamnus there, the little daisy flowered plant down there, have fully recovered now as a result of this project. Okay, right. So you may have heard of the Asian citrus psyllid, and I've been heavily involved in this program for, I can't believe it now, it's probably been close to 18 years now we've been working in various levels of this. So cottony, the Asian citrus psyllid, a well-known citrus pest, established in Florida. It was in Mexico. It spreads a bacterium that kills citrus, and that bacterium causes a lethal citrus disease referred to as huanglongbing or citrus greening. So you can see the adult psyllids here feeding on the plants. The young green stems of citrus can become highly densely infested with nymphs when they're not controlled. You can see the white honeydew ribbons coming out of the nymphs. In the middle photograph here, you see chlorotic looking leaves expressing the symptoms of HLB, and then eventually the plant will go into decline and die. Citrus production in Florida, where the insect and the disease are now widely spread, have caused a decline of at least 75% in citrus production in Florida. So California citrus industry had been watching the situation unfold in Florida. And then when Asian citrus psyllid was detected here in California, there was an immense amount of worry that California citrus industry was going to suffer a similar fate to what was being observed in Florida. So 2008, Asian citrus psyllid was found in San Diego County. It spread for several years. Massive insecticide spray programs failed to contain it. Then finally, the spray programs were wound down, and then we had permission to get funding to go to Pakistan, which is part of the native range of Asian citrus psyllid, and it has about a 70% climate match with citrus production growing areas in California, which is good for us. We want our natural enemies to be pre-adapted to the prevailing climates in California. So my wife Christina and I, we spent 3 years working in Pakistan on and off. We found 2 species of natural enemies, Tamarixia radiata and Diaphora incertus allegarensis. So in 2011, we began releases of the nymph parasitoid Tamarixia radiata. This occurred 3 years after Asian citrus psyllid had established in California. And during this time, the insect was spreading, and then the disease was being found in different counties within the state. Okay, so what, in retrospect, when I was going over this program, it was like, why did it take us so long to get started on this? It was obvious that at some stage, Asian citrus psyllid was going to come to California. We had pressure from Florida. There were populations pushing north out of Mexico. And as a biological control specialist, I felt I'd let the state down and the citrus growers down, and that we should have had these natural enemies already screened and pre-approved for release. So these ideas have been percolating in my mind for a while. So currently we've released about 25 million Tamarixia radiata. The populations of ACP have been decimated by these parasitoids, but that's a talk for a different day. So as I mentioned, there was about a 3-year delay in getting this biological control agent from Pakistan introduced into California, screened out of quarantine, and then the permission to release it. During this time, the psyllid's spreading. $5 million of insecticides are being sprayed. They treated about 6% of the infested properties in the LA area, and then it was just given up. A lot of time and a lot of money wasted. You couldn't contain the psyllid and you couldn't eradicate it. Okay. This brings me back to the thought that had been floating in my head. Should we have been better prepared for Asian citrus psyllid? And yes, we should have been. We already knew that ACP and the bacteria that it spreads were established in Florida, Texas, and Mexico, and I think it was inevitable that the invasion into California was going to happen. But we took the business as usual approach. We waited to see if ACP and the disease would establish. It was reactive. We waited, we watched, we assessed, lots of meetings to deliberate about what we would do, long lag in responding to this invasion, and that whole process took about 2 to 3 years. And during this time, ACP was spreading rapidly and the populations were becoming extremely dense in these urban areas. And I was wondering, well, you know, if we'd started earlier, maybe we could have cut this off. But because of the massive reliance on the spray program in the urban areas, the biological control program was not given permission to move forward because insecticides were thought to be able to contain and eradicate the insect that was the target of this program, ACP, but obviously that eventually did not happen. So I was thinking of doing this, well, maybe a disruptive approach is needed for dealing with these predictable threats. And this idea of proactive biological control that had been percolating around for a while started to crystallize. And I like to think of it as moving fast and fixing things before they become a massive problem for us. So the concept here is really pretty simple. You have your natural enemies approved for release before the anticipated invasion event occurs. I like to think of this as an insurance policy. You do all the work, you've got everything prepared, you hope you don't have to use your insurance policy, but you are pretty thankful that you have it should that disaster occur. You're ready to go very quickly. So I want to go over 2 adjectives here. The Europeans that have, and some of my New Zealand colleagues, which have also got enthusiastic about this idea, they're referring to this approach as preemptive biological control. I'm sticking to the adjective proactive, and I'd like to give you the definitions here. To be proactive means to control or mitigate a situation by taking action ahead of time rather than responding to the issue after it has happened. If you compare this to preemptive, the adjective preemptive, this means to prevent, eliminate, or deter an event from occurring. And a well-known military euphemism is a preemptive strike. You see the enemy on the hill over there, you fire a missile at them, you blow them up, they're destroyed, neutralized, and that threat has been eliminated. Preemptive biological control will not eliminate the threat of the invasion. All you are doing is getting proactively prepared for the potential invasion by having your natural enemies already lined up for potential use in the future. So I'm sticking with proactive, and I'm not enthusiastic about the term preemptive biological control. So, detection of the first established non-eradicable pest populations, you initiate your classical biological control program with the natural enemies that you've already proactively screened for release and that you've been given permission to establish. So this may require you to get those populations of natural enemies back again, get those colonies up and running in quarantine, and then you can begin releases very, very quickly because all the evaluation steps have been approved and you have your permits already to go for the release. So you could think of these releases being almost immediate and near real time for controlling these pest populations. So doing this actively ahead of time, I think, would potentially save you years of time in terms of evaluation and foreign exploration. If you can get your natural enemies out there while these populations are small and localized, you may be able to slow down pest population buildup and the rates of spread. You minimize invasion bridgehead risks. This is where you end up with pest populations budding ahead of the leading wave of the invasion front, and you get these small populations building up. And that's occurring because the mother population is so big and uncontrolled that you have these, like an amoeba, bits branching off and spreading into new areas. This becomes much harder to do if your populations are much smaller. And if you do spread into a new area, there's a good chance that the natural enemies will follow you and also provide some levels of control against those incipient populations. And then most importantly, you may minimize or reduce the adverse economic and environmental impacts that could be caused by the invasive species of interest. Okay, so the goal of a biological control program that was targeting Asian citrus psyllid was to bottle up and reduce these ACP populations in urban areas. And the idea here was to reduce the pressure of the insect and the disease moving out of these urban areas into commercial production areas. You wanted to keep the pests and the disease limited to urban environments. And for the most part, this approach has worked. There have been no detections of the bacteria that causes Huang Longbing in commercial citrus, and Asian citrus psyllid populations, for the most part, are very, very low, possibly non-existent in the majority of commercial citrus producing areas in California. So looking back in retrospect to what we have done with the biological control program with ACP, with the parasitoid from Pakistan, we've reduced those pest densities by more than 75% throughout Southern California. ACP and the disease are almost completely confined to urban areas, and we have attained this for more than 12 years now, perhaps closer to 13. This is the longest record ever attained anywhere where this insect and the disease have invaded. It's been kept in the urban areas, and so far it has failed to invade these commercial areas. There are other factors involved here as well. You know, there are quarantines and there are spray programs still going on in some areas. But for the most part, in the urban areas where ACP is largely confined, natural enemies are providing 99.98% of the control against that pest. So I'm predicting now there'll be no massive HLB epidemic sweeping through California citrus. If it does happen, I will be retired before that is realized. Okay, anyway. So I think the Asian citrus psyllid biocontrol program has contributed to this, but I also wonder if we had been proactive and had our Tamarixia approved for release before ACP showed up, could we have done even better? Reduce the incidences of the pathogen and the geographic spread of the psyllid and the pathogen throughout California? Could we have saved the state and the federal government millions and millions of dollars by eliminating the need for these spray programs that were targeting ACP in these urban areas? We'll never know the answer to those questions, but it's interesting to think about. Okay, so this graph that I want to share with you now shows typically what happens during an invasion process. The pest comes in, it builds up to high numbers, we release our natural enemies, and then we reduce the pest densities to low levels, which are now below the damage threshold. So no economic or environmental damage is now caused. But you can see there is a long lead-in time here, often years from when the pest was detected to when the implementation of the biological control program begins. It's reactive. What I'm suggesting that we do is that we step way back here before the pest is detected in California, and we initiate and complete a proactive biological control program before the pest of concern arrives in California. And then, during early detection, should pest populations be detected, you can begin a rapid reaction. You have a window of opportunity here to respond very quickly to that incursion with the natural enemies that were approved earlier during the proactive phase of this management program. And I wonder if the outcome could be something like this, where if the program could be initiated early enough, pest populations don't reach as high densities as you see here in the reactive program, and then the overall maintenance of those pest populations tends to be lower over time. This is all theoretical stuff. It's all word arguments with no numbers or any examples to back it up. But I find it an interesting thought experiment. So how would you go about selecting these targets for a proactive biological control program? Well, as I mentioned, you do these horizon scans for what we refer to as door knocker species. And I wrapped up a big project with the CDFA, probably 15 years ago now, where we looked at the invasive species that had established here in California. And what we detected was about 44% of those invaders established in California from other areas of the United States and Canada. So they were already present in the larger United States, and they were working their way to California. So if we did the horizon scans and looked for these species, we should have been prepared for many of them that were coming. So for example, Asian citrus psyllid, established in Florida in 1995, Texas in 2001, showed up here in California in 2008. Brown marmorated stink bug, established in Pennsylvania in 1998, showed up 7 years later here in California in 2005. South American palm weevil, established in Tijuana, Baja California, Mexico in 2010, established in California in 2014. Spotted lanternfly, established in Pennsylvania in 2014, working its way westwards to California. The question now is, when will it arrive and where will it establish first in the state? Other areas you can think about are pests that may be associated with agricultural, horticultural, floricultural imports. These are known conduits for moving live pests through trade into different parts of the US and California in particular. USDA APHIS has Pest Lens, which is an online newsletter you can subscribe to, which provides early warning notifications about first finds of pests, not only in the United States, but in other countries around the world as well. This may give you a heads up as to what's moving globally, which could potentially come into California. And of course, surveillance interceptions. CDFA is doing this a lot. They may be trapping pests of concern, but those populations are not detected. So for example, false codling moth has been picked up in traps in California. This is a notorious agricultural pest. If live adults are being detected in pheromone traps, then maybe it's just a matter of time before some of those live adults establish populations in agricultural areas or maybe even urban areas, most likely, and this is potentially a pest we should be concerned about. And then there is the identification of what I refer to as wild cards. These may be potential pest species about which we know nothing, and they suddenly emerge and start moving globally, and they come to California, for example, and we don't know much about them before they show up and become problematic. These are probably almost impossible to identify ahead of time. So, has proactive biological control ever been attempted before? Yes, it has. So one of the first programs was run out of my lab where we were interested in looking at fruit feeding moths associated with fresh avocados that were being imported from areas where avocados are native and where the natural biodiversity of fruit feeding insects associated with that fruit is very high because it's the evolutionary area of origin in parts of Mexico, Central America. But almost nothing is known about those pests. So we've done these proactive projects in Peru and Guatemala. We looked at the pest species associated with those fruits. We developed natural enemy inventories. We developed life tables, and we identified and field tested the sex pheromones for some of those insect pests. This was to prepare California ahead of time for these potential invasive species that could be associated with fresh fruit imports out of these countries. We discovered several new species of moth that were associated with avocado fruit. Nothing was known about them. They'd never been described before. These are the wild cards that I was talking about. But now we are better prepared for those wild cards because we've done this footwork, groundwork in these countries where fruit is exported from. New Zealand has taken a pretty bold approach here. They're very worried about brown marmorated stink bug being accidentally imported into New Zealand and causing a lot of problems for their agricultural and horticultural industries. And they have already screened and had approved for release the brown marmorated stink bug egg parasitoid Trisulcus japonicus. And this was relatively easy for the Kiwis to do because they have a very impoverished native stink bug fauna, and several of those native species live up in the mountains in alpine areas where it is theoretically unlikely for the brown marmorated stink bug egg parasitoid to move into. Okay, so what's California doing about proactive biological control? Well, back in 2018, we put out an opinion piece that was published in California Agriculture. This got some traction within the CDFA. And the CDFA's Office of Environmental Farming and Innovation and Pesticide Consultation and Analysis is actually supporting proactive biological control and IPM programs that are focusing on pests that have potential to come to California or have very recently established here in the state, and we need a rapid response to managing those pest problems. So calls for proposals have been made annually since 2019. These tend to be 3-year grants, which range from $260,000 up to $500,000. This program has made a huge difference in getting prepared for these invasive species not only with the natural enemies, but learning a lot about the biology and behavior and possible control strategies for these pests in areas where they are causing problems, in areas where they are native. We can use all that information, we can collate it and have it ready for use for California in advance of those pests coming into the state. So some of the projects that have been worked on have been spotted lanternfly, avocado seed weevils, Tuta absoluta, swede midge, emerald ash borer is now on this list as well. And if you're interested, you can go to this website to look for details on this. Okay, so some potential targets. We have been spearheading a program against spotted lanternfly. This insect is native to India, China, parts of Vietnam. Showed up in Pennsylvania in 2014, rapidly moved up and down the eastern seaboard, causing big problems in apples, massive problems in grapes. Has been associated with the death of grapevines in Pennsylvania. Also, when the population densities of this pest are high, it can get in tomatoes and corn and cause quite a few problems. It is an indiscriminate egg layer. It will lay its eggs on train carriages, for example. It will lay its eggs on pieces of outdoor artwork. And not last year, but the year before, at the border stations, CDFA picked up a large piece of artwork at the outside that was being moved into the state that was covered in spotted lanternfly eggs. It was sent back to Las Vegas to be cleaned up. They've found spotted lanternfly eggs on loads of firewood that are coming into the state. There have been no detections of this pest in California as of yet. By that I mean populations of it, but it has been detected at the borders, which suggests there is pressure coming into California, which may also suggest that it's just a matter of time before this pest successfully invades and establishes here. It's highly prolificous, threatens several agricultural commodities. You can see high density populations of spotted lanternfly on these grapevines that are growing in Pennsylvania. Is biological control an option? Well, we thought it was, and we did a lot of work with our colleagues with the USDA on the East Coast. They evaluated an egg parasitoid from China, Anastatus orientalis. They looked at native East Coast species. We ran a similar program here looking at native Western species that the egg parasitoid could attack. And unfortunately, it is an extremely prolificous parasitoid. It will attack a lot of different species of native insects other than spotted lanternfly, including important native moth species. So we have determined that this natural enemy from China has too broad a host range. It's not host specific enough and will not be considered for use in a biological control program in the United States. Okay, so this sounds a little disappointing, which it was obviously, but what we learned from this is that we now know that there's no point spending a lot of time and a lot of money on evaluating these biological control agents because they won't be approved for release. Those monies, those funds, that support can be put into other control efforts. Maybe it'll be detection and eradication, for example, and we will not lose time with a biological control program that is unlikely to be approved for use in the United States. Okay, so in conclusion, our invasive species, pest species issues, they're not diminishing. And here, we're dealing with South American palm weevil. I've been working in Brazil where there is a parasitic fly that attacks the pupae of this palm weevil, and this fly may have potential future use against this pest in California. I think the business as usual approach, reactive, classical biological control, I think we need to disrupt this. Let's get prepared early. Let's do horizon scans for door knockers. Look for emerging invasive pests. These can be weeds or insects. And maybe even through these foreign exploration efforts that we do overseas, maybe we can identify more wild cards, species that we know nothing about, but are obviously associated, say, with a commodity like avocados that get imported into California, and we can identify these new threats ahead of time. I think proactive biological control really is a new spin on a classic approach to managing invasive insect pests with host-specific natural enemies. So, I don't know, probably nobody's interested in reading this article. I'm probably the only person who's read it because I wrote it. Well, we've put this idea out here. Here's a new paradigm. Proactive biological control of insect pests. It was published in the journal Biocontrol. That's a very elegant idea. It's simple. It's common sense, I think. That's what I love about this concept. I would say that in terms of biological control, this idea is probably equivalent to Einstein's E equals MC squared. Small but powerful. What do you think of that, Albert? Yeah. All right. And getting a hand clap from the man himself. All right. That's all I have. I hope you liked my joke. And if you have any questions, yeah, let's discuss proactive biological control.
Our first question is the individual wants to know if South American palm weevil has been eradicated in California.
No, it has not been eradicated. The populations are high. It is spreading slowly, and we estimate it's probably killed over 20,000 palm trees in San Diego County, but still largely confined to the southern part of San Diego County. It's now established around Camp Pendleton, and it's likely to push over the top of Camp Pendleton and keep moving its way northwards. But it's an extremely slow-moving species, and I think part of the reason for this is that there's just so much food for it. There's no motivation for it to fly long distances and establish these populations ahead of the leading edge of the invasion.
Thank you. Next question, if biocontrol is extremely host specific, won't the biocontrol agents suffer or die off without having its host available?
Yeah, that's a good question. It's one I get asked often. And typically what happens is that these natural enemies will not cause the extinction of their target or preferred prey. They just go to very low densities. So an example I'd like to give of that is what we're dealing with now with Asian citrus psyllid in Southern California. Some of our study sites have not had psyllids for over 4 years now. They're completely gone. But rarely, we will find small populations of Asian citrus psyllid that has the Pakistani parasitoid associated with it. So one way of looking at this now is that the pest and the natural enemy are kind of playing like a game called hopscotch throughout the environment. Asian citrus psyllid may escape and get a little population going over here. Then the psyllid parasitoid finds it, knocks it down. Maybe a few of those will escape and start a population somewhere else, and they just end up sort of moving around the environment like this. But the levels become very low. They're no longer problematic, but we have not caused the extinction of either the target pest or the natural enemy itself.
Thank you. Spotted lanternfly, would a control mechanism for it be the elimination of host plants such as Ailanthus, which is also known as tree of heaven?
Yeah, that's a great question. And yes, I think taking out its preferred host plant, tree of heaven, which is also a preferred host plant for another invasive pest, brown marmorated stink bug, which is also native to China, is a very good idea. I think this cultural control technique has been emphasized in the major grape production areas of Northern California, where the wine industry could be at risk, is to start taking out ahead of time tree of heaven plants that could enable spotted lanternfly to establish in those areas. You know, surveillance coming across the borders, like the border control guys were doing up at Truckee, where they detected those 2 potential incursions of egg masses, very important. I suspect it'll be egg masses that's come through into California, maybe on somebody's RV, barbecue equipment, firewood, and then once those eggs are introduced into a particular area, those nymphs will hatch. Spotted lanternfly is extremely prolific, and it's likely it has a good chance of finding a host plant it can survive and reproduce on, and that'll allow the populations to get away. And I suspect also it's likely that that will happen in an urban environment as opposed to an agricultural production area.
How can the public get more involved with these issues of being proactive, especially with regards to imports and invasive species coming in from outside of our borders?
Yeah, so that's a great question too, and this is one I struggle with a lot. Seminars like this obviously help a lot, but we're reaching what, maybe 105 people today? And we need to reach tens of millions of people in California about these problems. So communication, outreach, and extension like we're doing here is obviously very important. Another thing that I've noticed that I think has declined is when you now come into these international airports like LAX, for example, questions about bringing in fruit, meats, you know, honey, other stuff like that, that's pretty much fallen by the wayside now. And I don't know what's going on behind the scenes, but seeing this reduced interest in whether or not people have things in their suitcases that they should not be bringing in is of concern to me. So I've just come back recently. I get asked, do you have anything in your bags you should declare? And I just say no. They go, oh, that's fine. Through you go. So you have to wonder, with all the fruit fly outbreaks we've had in Southern California last year, I think we were battling maybe 7 species at the same time. I mean, that's unprecedented. It's never happened before. The only way those things are getting in is through infested fruit. And that infested fruit, in my opinion, is likely to have been inside people's suitcases that have been moved from different parts of the world. They get here, they open it up, that mango's all rotten, it's full of maggots, and you just toss it outside and boom, you get these small fruit fly populations building up, which now need to be not only detected, but maintained and contained and eradicated. So there's a lot of education that needs to go on. That's definitely important. But money also needs to be invested at the border. And we talked about pre-border, border, and then localized post-border inspections to try and manage these small populations before they really get out of hand. Got to detect, contain, and eradicate if possible.
Thank you, Mark.
We're done.
All right. I just wanted to note we're just past the 1 o'clock mark.
Sorry.
So if people need to leave, you can. Mark, are you able to stay?
I can stick around until 1:15, then I have a meeting with the CDFA at 2 p.m., but I have to drive off campus to get to that at Mount Rubidoux.
Fantastic. So if anybody needs to leave, thank you for joining us today. Thank you, Mark, for
you're welcome.
a phenomenal presentation.
Oh, thank you. Thank you very much.
Lots of good interest, good comments in the chat complimenting you.
Thank you.
And I'll just turn it back over to Julie for additional questions.
Okay.
Thank you. The next question inquires if there are biocontrol agents being investigated or available for invasive shot hole borers.
Oh, yes. So that work has been conducted by the Stouthammer Lab. Richard's retired now. It's been carried on in part by Paul Rugman Jones, who was a research associate with Richard's lab. So in Taiwan, they've greened, I think it's mainly looking for parasitoids that are associated with the larvae and pupae of shot hole borer. I think they've found some, but my feeling is that they're maybe not overly promising to provide a lot of control of shot hole borer. And I think this in part is just due to the biology of that insect. It lives in those tunnels. Brothers and sisters mate in those tunnels. They come out for a very limited time before they bore new tunnels and go back in to the tree again. So any natural enemy would have to be really good at working its way through those tunnels to find the brood that would need to be attacked. And not all targets, invasive species, are good targets for biological control. Some pest groups are better targets than others because they have efficient natural enemies associated with them. So for example, thrips, in my opinion, are very poor targets for classical biological control, whereas something like a whitefly or a mealybug may be a much better target because they have a richer suite of parasitoids typically associated with them.
Thank you. This next question, the questioner would like to know about the roles of collaboration between different states that have invasions, like ACP in California and Florida.
Right. Yeah, so I had not been involved with anything to do with Asian citrus psyllid until it came into California, and there was obviously a need to do something about that problem after the insect arrived here. So I don't know what was going on between California and Florida and Mexico up to that point, so I can't really answer that question. But I can provide some insight into how we've been dealing with spotted lanternfly. And with support from the CDFA's proactive biocontrol program, we were able to get involved fairly quickly with the spotted lanternfly program with collaborators on the East Coast. And those collaborators were at Penn State and also with the USDA ARS, who were leading the foreign exploration efforts in and around Beijing, China, where spotted lanternfly populations in the US originated from. So at a very early stage, we opened up those networks of communication. We got funding, thankfully, that enabled us to start this work. And USDA was very supportive of this East Coast, West Coast program operating at the same time. And they supplied us with the egg parasitoids from China so we could run our own evaluations out here in the western parts of the United States. So if those communication channels had not been open, if we did not have good relations with colleagues in other parts of the country, and obviously we didn't have the funding to support this work, then that program probably would have got nowhere. So yeah, I think maintaining good relations with these agencies, with your colleagues, with the funding agencies, doing good job, getting your reports on time, demonstrating that you can deliver the results that you've promised in those grants has really helped the spotted lanternfly project. And obviously I'm disappointed that the natural enemy didn't work out how we had hoped, but as I mentioned in the presentation, this is really important to know ahead of time before we spend 3 to 5 years post-invasion trying to figure that out. So we're already ahead of the game.
Have you ever do you ever observe negative effects of introduced biocontrol agents on native arthropods?
Yes, so there are some documented cases of that. I'm not aware of any in California, but on the east coast of the United States, yes, it's been well documented that gypsy moth parasitoids that were imported in the late 1800s through maybe to the mid 1900s, 1950s, 1960s, something like that, definitely have had a negative impact on native Lepidoptera. And one species of fly in particular, Compsoglyra consonata, it's a parasitic tachinid fly, has very broad host range. It attacks moth larvae, attacks some species of sawfly larvae as well, which was released for the control of gypsy moth, has had a devastating impact on saturn-eared moths and those big, beautiful green lunar moths. But that impact of that parasitic fly is not consistent throughout the whole native range of gypsy moth, and other factors have influenced the decline of those big, showy moth species as well. Now, this shows, I'm just going to use this as an example to illustrate how the philosophy of biological control with respect to natural enemy specificity has changed. So when these natural enemies for gypsy moth were imported from Europe into the eastern United States for the control of gypsy moth, which builds up to massive populations and defoliates hundreds of thousands of acres of forest maybe, it was thought, well, generalist natural enemies that attack not only gypsy moth, but can sustain good populations on non-target species could be really good for us. Because as soon as gypsy moth outbreaks occur, we already have a large standing crop of natural enemies that have been living on native species and non-target species, and they can immediately move over onto the gypsy moth outbreaks and suppress them quickly. That was the theory. And there is some experimental evidence to suggest that that parasitic fly, Compsoglyra consonata, yes, it does maintain a large standing population during non-outbreak years, and yes, it will respond very quickly to at least artificial gypsy moth outbreaks that research scientists create in the oak forests over in Massachusetts, for example, where I did my PhD and was familiar with this work. As we move into the 80s and 90s, there's been a large shift in our thinking about native species, and that maybe they're really quite nice and we should be doing more to protect them. And that has led to a shift to reducing our reliance on generalist natural enemies and becoming more focused on finding natural enemies that have a very narrow host range and high levels of specificity, with the goal of not only ensuring you've got a good targeted biological control agent, but you're going to reduce or eliminate, mitigate the collateral fallout against these non-target native species.
Thank you. Do you know of any watch list pests for avocados?
Yeah, I do. Yes, so we're quite concerned about, especially with fresh fruit imports, mainly weevils and moths that are associated with avocados. And as part of this proactive biocontrol IPM program that the CDFA runs, we've been looking at natural enemies and finding the aggregation pheromone for the avocado seed weevil that could be a potential future threat to California.
Does red palm weevil have the same natural enemies as South American palm weevil?
Yes, it does not. As far as I'm aware, and I've worked a lot in Southeast Asia on this with colleagues in Indonesia, parts of Thailand, Malaysia, the Philippines, Vietnam, we've found no evidence of parasitism against red palm weevil. However, the levels of parasitism against South American palm weevil in the northeastern parts of Brazil appear to be quite high. We suspect that that fly is probably not specific just to South American palm weevil, but it is likely to attack larvae of other species of Rhynchophorus, like Rhynchophorus ferrugineus, which is the red weevil. South American palm weevil is Rhynchophorus palmarum. And there's been some interest, at least in the Middle East, about getting this parasitic fly out of South America, Brazil in particular, the only area where it's found, and for it to be evaluated as a new association. Oh, maybe I should add that to my list. A new association biological control agent targeting red palm weevil in areas where that pest is invaded. So maybe around the Mediterranean, the northern parts of the Maghreb region of Africa, and then, of course, the Middle East, where dates have been quite severely impacted by that pest.
This is a question the questioner would like to know how land managers can access, order, request biocontrol agents that have been approved. The example used was for controlling an invasive plant, but nonetheless, I would think that they're looking for guidance.
Yeah, so this is a tricky one. A lot of these natural enemies are not commercially available, so you would need to plug yourself into a program that is currently running targeting those invasive weeds that are of interest for you. So for example, in Colorado, they have agencies that collect, reproduce, and then redistribute some of these weed biological control agents. The CDFA has a weed management program. USDA ARS has a weed biocontrol lab running out of Albany, up near Berkeley, California. So those are agencies you may be able to contact if you have a particular weed and a specific natural enemy that's of concern to you. So when it comes to the insects, yeah, so say like, I don't know, what could be, say gold spotted oak borer, for example. We don't have natural enemies available for that pest. And often these programs, after they have been initiated and they cease, colonies of those insects are not kept. And basically you're just relying on the natural enemy to sustain itself and then to distribute itself naturally through the environment. But it's also possible that you could go into those areas, collect those parasitized targets, which could be hard to do, probably take a lot of time. There may not be a lot of them around to find. But then you would need permits from the CDFA to relocate them into new areas of California. Yeah, so it can be a little complicated.
Well, that was our last question, and it's 1:15. Randall, you want to wrap it, please?
Well, I again think this was a phenomenal presentation, and you're getting some great feedback here. And so we'll let you go on to your next task, but thank you very much for being here today.
All right, you're welcome. Thank you, everybody, for your time. And thank you, Randall and Julie, for helping arrange today's meeting. I'm always happy to help with these. So thank you very much for that.
All right, take care.
All right,
Thank
see you guys.
you. Thank
Bye-bye.
you, everyone.
Good afternoon, everybody. Bye-bye.